Everyone, thanks for joining us for the June seminar in physical genomics. My name is Benjamin Keene here at Center for Physical Genomics at Northwestern University. We're really excited for today's seminar. It's something a little different. Um, we've got our alumni, Dr. Vasundhara Agrawal. She did her PhD in Biomedical Engineering here at Northwestern, and Professor Vadim Backman's lab, who is the CPG director. Her work at Northwestern specialized in imaging and biophotonics. She worked at the intersection of chromatin biology, biophysics, and imaging, developing methods to study the three-dimensional organization of the genome. Her work integrated imaging, computational analysis, and physics-based statistical approaches to elucidate genome structure and function with applications in regenerative engineering, among others. She's now applications manager for spatial biology and image analysis at Leica's Microsystems at their Boston Innovation Hub. She works at the cutting edge of multiplexed whole tissue imaging, AI-powered image analysis, and spatial statistics. Her work focuses on uncovering molecular signatures across complex biological systems, with particular emphasis on the tumor microenvironment and cancers of diverse origins, as well as neurodegenerative diseases such as Alzheimer's. As you can tell, it's a mouthful for me. Vasundhara, very impressive. It's great to have you back with us. So for those of you in the audience, Vasundhara is going to talk today about her path from academic PhD research into industry. I know a lot of our audience are trainees themselves, so this is kind of a topic we don't often get to address with these talks. So hopefully you guys will take something good from it. We're really excited. Vasundhara's great. If you've got questions, feel free to type them into the chat at any point, and we'll address them at the end, or just raise your Zoom hand or turn your mic on at the end, and we'll do the Q&A then. Feel free to check out our website, social medias at CPGE. We've got a lot going on, so follow us. And with that, I'll turn things over to you, Vasundhara. Thank you so much.
Thank you, Ben, and thank you, Sri, for organizing this. And I want to thank CPGE for hosting me today. And today I will be speaking about academia to industry and how to think about the boundary. The views here expressed here are my own and do not necessarily reflect those of my employer. And I think what I want to do first is just to go through my background so that it gives you some perspective into the kind of decisions I made throughout my scientific journey. So about me, I started off in 2013 at IIT Chicago, which is not too far from Northwestern. I worked during my undergrad in three different labs, one in electrical engineering, the next one in chemical, and then finally I ended up working in a biomedical engineering focused lab that looked at cancer imaging, led by Professor Kenneth Tashauer. And my emphasis for my bachelor's ended up being in cell and tissue engineering as a track in biomedical engineering. So around 2017 is when I joined Northwestern. I was a doctoral student in Professor Vadim Backman's lab, where the main focus was on looking at chromatin conformation and its influence on cell fate. A lot of my early years were focused on training on different imaging systems and also understanding transcriptomics datasets and analyzing these two kinds of datasets with an application towards cell engineering for improving regenerative outcomes. So one driving question throughout my PhD and most of my academic life has been cells with identical DNA. So they have the same genomic DNA, but they perform very differently. So they have different functions or different phenotypes. And how does that happen? So that was kind of the driving influence throughout my doctoral research. And around 2019 is when Center for Physical Genomics and engineering was inaugurated. Around 2020 is when there was peak COVID. So naturally, it became slightly harder to collect data, especially if you were collaborating with different labs. So my emphasis became more towards larger data sets and analyzing those large data sets. So I took a bunch of courses in Python, on Udemy and Coursera, and those really helped me to understand how is it that I can analyze these large data sets. It also made me understand the importance of working in a team. I think our lab did a fabulous job in coordinating how to work together in such a tough time, because it was difficult to coordinate how to do the experiments in those circumstances when COVID had was really at its peak. So it was a tremendous challenge, but it also gave us a lot of opportunities to, let's say, work on analyzing these data sets that we've already collected and work towards publishing those with an interest to understand mechanisms. And around 2023 is where when I graduated, and I had this growing interest in spatial biology. I ended up joining Leica Microsystems, and my role was based in Boston, Massachusetts, so I moved there. So this is kind of just a brief timeline, and now I'll go back to talking more about the question, that underlying question that made me interested. So different cells in our body have the same DNA, yet there's a very specific gene expression program for a given function. So what that means is that cells decipher the same DNA sequence differently, which determines their function or phenotype. And this kind of helped me understand also the process of differentiation. So when a really plastic cell becomes something so stable, depending on which pathway it takes, and it can be thought of as a Rubik's Cube, where you can modulate this transcriptional reprogramming and alter the state of the cell to modify the function, the ultimate function. So what this means is that there is the genetic code, so the DNA sequence. On top of that is the epigenetic code, so the modifications that can influence which genes are expressed. And then in the z-axis is what I think of it as the 3D chromatin packing, or how the DNA, the proteins, the RNA are together in this space, in this small nucleus. And you can look at the spatial positioning, the folding, the dynamics, and how it influences which genes are expressed. So this was kind of what most of my work in my doctoral research was focused on. Basically, first measuring how chromatin is altered at different length scales, and then engineering it in a system where I can see whether modulating the chromatin structure alters the cell fate, and then deploying it in analogous systems in biology. And how I did that was in different parts. So we had a fantastic toolbox in our lab that allows us to look at chromatin, and I'll show a few images to give you an idea of what that looks like. And then my focus for the application was on cell engineering to improve regenerative outcomes. And the last part was to look at other avenues where this system could be deployed. And how we did this in the lab was with high resolution electron microscopy. So here is a chrome stem image shown on the right, and you can kind of really see all these smaller features that you would not be able to see with other imaging techniques. So the higher resolution really allows you to look at the chromatin domains and structures like the lamin-associated domains, and even finer structures like the nuclear envelope. And you could do this in 3D, so you can collect the images of these thin sections that you make of your slide or your tissue culture wells, and put heavy metal dyes on them that bind to the DNA, and then look at it with an electron microscope that allows you to go at this high resolution and see how the structure of the DNA is modulated in different cell types upon a treatment versus in a control. And not to go into too much detail, what was truly fabulous was to be able to look at this approach of chromatin, the transcriptional patterns, and the phenotype through these different lenses. So there was the modeling approach of looking at transcriptional processes, the nano imaging that allowed you to look at chromatin at different resolutions, and then the genomics aspect where you could connect the conformation and the transcriptional patterns through sequencing-based approaches, as well as the imaging ones. And finally, the engineering, which is where most of this mechanistic study lies to really regulate the chromatin structure and relate transcription and phenotype. So essentially, this idea of looking at chromatin could be applied in these different models. So, for example, one of the aspects that I focused on was looking at targeted differentiation, shown here, and really enhance the efficiency of a cell type, differentiating into your final cell type of interest. And this was a lot of this work was done with Professor Guillermo Amir's lab at Northwestern. And then also looking at the enhancing adaptive potential and seeing how cells that are exposed to stress, do they have certain modifications in their chromatin, and could those be engineered to make them even more resilient if they face a different kind of stress in the future? Uh, and one thing to note about these models is that there's this analogy going from the adult cell state to a stem cell-like state, which is more plastic. You could pose similarities in different types of models, such as senescence, reprogramming, cancer stem cells, cells surviving upon exposure to stress. And one topic that I was particularly interested in was understanding senescence. So what happens with aging is that there's typically a despecialization of adult cells, so they stop performing the job that they're supposed to, and these senescent cell types start getting accumulated, which have these changes, such as increase in nuclear size, loss of lamin B, V1, increase in RNA Pol II elongation speed, large heterochromatin foci moving towards the nuclear interior. And basically, what I want to emphasize is that there were multiple markers to first identify and confirm the presence of senescence, and this differs in different cell types that you look at. So depending on the tissue, these markers could be different. And that made me realize that the work that I did in my PhD was more focused on looking at the single nucleus. So looking at these techniques, looking at 3D genome with chromostem and nuclear mechanics, and how the genome conformation shapes fate. While in the industry, I now look at things at the whole tissue level, and I'll show that in a bit more detail in the next few slides. But what I'm doing is that I'm looking at multiplexed imaging, so not just one marker, but multiple markers at the same time, and doing tissue analysis at scale. So similar question, but the lens is a bit wider. So at Leica, what I'm currently working on is these multiplexed imaging called cell dive, where you can basically iteratively stain multiple markers. So here, what you see is within one round, you can look at 4 different immune cell types, and then you can iteratively keep adding more markers. And what you can then do is essentially create a map of your uh, tumor architecture and label it with as many markers as possible. And look how those cells are different based on their geography. And this is particularly important for, let's say, the field of neuroscience and cancer, looking at the tumor microenvironment, where you essentially want to look at not just one marker, but how multiple markers are changing at the same time. And what you can also do with spatial biology is look at different structures depending on how zoomed in or zoomed out you are. So at the smallest level, you can look at cell composition, so identify immune, tumor, stromal markers, all sorts of combinations of those, and then how these cells are interacting with each other, and what the microenvironment looks like, and finally the larger microanatomical structures. So essentially, it reveals details about the tissue at different length scales. And you can apply this through different tumor models. So what you see here is a very zoomed out view of the whole tissue. And you can see how large it is based on the scale bar. But you can look at colon carcinoma, lymphoma, breast carcinoma. These are just a few examples. And what's truly unique is you can not only look at the immune cell subtypes. So, for example, here you see cytotoxic and helper T cells. And at the same frame, you can look at other cells, let's say B cells, and you can look at all of them together. Uh, and some of my work that I do is identifying how these spatial patterns reveal, uh, let's say, rare cells or unique information about a particular tumor. In here, you can see GLUT1, which is a hypoxia marker, and how a specific zoomed in region of the cell shows how all these other markers are changing in these GLUT1 enriched regions. So you can ask these kinds of questions that are there any immune cell types that are more enriched in these highly hypoxic regions of the tissue? And it truly lets you look at these things in more detail. So here I'm doing more of a clustering based on expression of GLUT1 and identifying regions that are highly, medium, and lowly expressed for this marker, and then identifying how all the other markers are changed in these regions. Um, and this truly helps you figure out, are there specific cell types that might be more enriched in these hypoxic regions? So here I'm showing that, let's say, M2 like macrophages and dendritic cells are more, you have a higher probability of seeing those in these GLUT1 positive regions. And again, you can do more of these spatial analysis to identify hubs of cells in these regions based on, again, the XY location, and even Z if you're doing it in 3D. So this was just to give you an example of what kind of work I did in the lab and then what I do now. And what I want to emphasize is that the science as the constant. So the lens can change depending on which institution you go to, but the question that I was interested in remained the same. And now I think what I want to think more about is if we think about the boundary. So around the time when I was closer to the end of my PhD, I started thinking about what I wanted to do next. Um, One of the books that I particularly found helpful was this book called Designing Your Life. One of my mentors had recommended it to me. What it does is that it uses the design thinking to prototype and test. So you basically write down a bunch of ideas, and then you write them down in a lot of detail and then you test them out. But I'll go over what I mean in a bit. And essentially, what I was doing at the time was I was talking to others who had done it. So we had in our lab, there have been a lot of really intelligent students who graduated, and talking to them who are already, let's say, in the industry or in academia really helped me because I could ask them a lot of questions. I could get a sense of what their day-to-day was like. And then wherever I thought I was still confused, I could seek advice from mentors who were a little were ahead of me, who had already graduated a long time ago. So what this book really helped me do was it helped me identify, what my reality was and identify gravity problems. So, what does a gravity problem mean? It means something you cannot change. And uh, so, I would give an example. For me, it was, let's say, my visa situation. Because I was an international student, there were some roles that were not open to me. So that would be a gravity problem, something that I cannot change, but I can maneuver around. So it's good to know, let's say, what your gravity problem is. Are you looking for a job that's in the same location? Let's say something that essentially you should mention to your employer that this is something, it is what it is. And then you can think about the plans based on that. And then the second one would be building your own compass. So basically identifying what your values are, and what about the work that you value, uh, and how your life should be. So I think this was really important for me to do, and I'll also explain how I got here. And then the third one was basically ideating multiple lives. So what it says is that you should sketch out three very different trajectories of how your life could go. One, if you continue doing what you were doing and how that looks like for the next 5 years. Two, if let's say you couldn't do what you're doing right now, how would that look like? What would you be doing instead? So lay out those 5 years. And third was if resources were infinite, what would you do essentially? So basically, if you write those down, then you have a little more clarity about, let's say, if you're not sure about what you want to do, you at least know what kind of questions you can discuss with someone who's already in the field. So I think these kinds of early exercises help you talk to your mentors or identify mentors who might be helpful to you. So another really important thing that I would like to mention is there were several programs at Northwestern that helped me. The first one would be the Northwestern Externship Program. It used to be called NU Next. What that allowed you to do was it matched you with different uh, people who had graduated from Northwestern who are in, let's say, who are doing something that you would like to do in the future. If you are interested in going in biopharma industry based job, you could be matched with three or four uh, uh, specialists in that field, and you can schedule meetings where either you can have your resume reviewed or they can walk you through a day in their life, help you understand what is it that a day-to-day looks like in their industry. So I found it extremely helpful. I did a few that were more about consulting because at the time I was interested in pursuing a role in consulting based on the kind of conversations I was having. And then two other programs that also helped me was there was the Northwestern Prison Education Program and the Northwestern Mentorship Opportunities for Research Engagement, so the MORE program. These were more focused on teaching and mentoring, and they made me realize that I really enjoyed both of these aspects of science. And you could essentially design your own course. So I highly found it valuable. Um, and these are, I think, the kind of programs that really help you narrow down your interests, because you might be starting off with a lot of interest, but these help you figure out what is it that you like. So without trying, it's hard to say that I'm interested in something. The next one I wanted to highlight was the Center for Leadership. So this was a program that had a fellowship for grad students and postdocs. And what this program did was, it was, I think, a few months long, and there were different books and readings that were assigned, followed by activities that you did with an instructor. And it helped me understand what my strengths were, because the whole idea was to follow strengths-based leadership. So knowing what you're good at, you can hone on your skills instead of focusing on what you don't have. And what I understood was that as scientists, we tend to have a lot of strengths in the execution area, but sometimes not so much in the strategy versus influencing and relationship building. So I think it was good to identify what is it that I need more work in, and what is it that I can continue developing. Another thing it did help, oops, sorry, was what are my values? So I still have a notebook where I wrote them down depending on like 3 or 4 exercises that were done. And I identified that these 5 were my values: creativity, quality, helping others, expertise, independence. So I think those really helped me decide, let's say, what would be the right opportunity for me, depending on what shows up. And just want to mention this quote again: if you do not know which port you're seeking, no wind is favorable. Um, so I think it's good to write down what is it that you like doing, and then narrow down your options from there. So next thing I want to talk about skills that transfer and skills you build. So from my PhD, I think one of the most valuable skills I would say was the framework building, which is thinking about a topic broadly as well as deeply. So being able to zoom in and out across scales, literally like a microscope. Um, you can think about this as a review paper versus a mechanistic paper. So a review paper is more broad, a mechanistic paper is more specific to a pathway or what your hypothesis is. And I think that skill really helps you have conversations with different types of people. So if you're more in uh, a very heavy scientific discussion, you should be able to talk very deeply, but then also take a step back and think about the big picture. And some of these other skills that were scientific judgment, deep reading, and pattern recognition. But what I had to build was, I would say, the most important one was efficiency. So this is something that is focused on it's basically focused effort and getting things done in a very short time, let's say. Not even short time, but by a specific deadline. That I think was new to me. And also communicating and collaborating with a very diverse group. I think that's something you still get exposed to in your PhD, but it is you also have to think about not just the science, but also the big picture, like I was saying before. So my core PhD lessons, I would say, during this time were looking at the intersection of values, skills, and the level of difficulty. So I, I think if there's a good mix of all these three, then it makes sense. Like if something aligns with your values and you have the skills for it, but it's just too easy, then maybe it would make you bored very easily too. So you have to think about that as well. And I think to go over these main points, I want to say that the first one, I think, is really important to be able to break down problems to its simplest building block. So to identify the root cause. And I think this is the most important lesson from my PhD, to not go into the more complex things first, but go to the basics. Always go back to the basics. If you're looking at a transcriptional process, go back to the very basics of how the biology and the process of transcription works, and then look at the more complex structures. The second one would be plan and have a mitigation strategy. So I think e- I'm sure with all of us in the room who do experiments every day, your experiments don't always work, um, even if you've made the best plan. And I think that's why it's always important to have a backup. So if, let's say, a really critical experiment that you need to get something done is not working, I think you should be able to plan for it even before you start this experiment, because it really helps you save a lot of time moving forward. And even just thinking about resources, I think it's a good way to manage your resources and where you should spend it before you do like a big experiment. And then third one, spending more time on what you enjoy and what comes naturally. I would really think about, like, while you're in your PhD program, which aspect do you enjoy the most? It could also be something as simple as making the presentation for your lab meeting and presenting those results. I think for me, that was my favorite part. Um, and it may not be the favorite part for a lot of people. Um, Also, another favorite part was really talking about my ideas with other scientists and hearing what kind of feedback they had. That's where a lot of learning happens. So I think that's to think about as you're doing the work, what you like and what you really don't like would help you understand what is it that you want to do more of. And fourth would be learn from people who have done it before. So having conversations with other scientists, either in academia or industry, whose work you like and you've been following, I think that's really helpful to understand their perspective. A lot of times it might be very different from yours. You don't have to take all the feedback because feedback is like a gift. So you see by talking to a lot of people, you'll get a sense of what kind of environment you want to work in. And then fifth, the only way to find out if you like something is to try it. Um, so I think there's what I was mentioning with the different programs at Northwestern that helped me. I think it's really important to be able to try different things because otherwise it's hard for me to say, let's say, I don't want to pursue consulting as an option, as a path after my PhD. I can't say that without really giving it a shot. So I think there's a lot of value in doing things while you're working towards your PhD to figure out that, okay, this may not be the right path. Um, so I think this is a good summary of kind of what I wanted to cover today. Um, Oops. And I wanted to thank our Center for Physical Genomics and Engineering again. This is one of the summer barbecues that we had in 2021, I think. And then the image on the right is from 2023, right about the time I graduated. So it's really I think your PhD are truly the one of the best years of your life. So you should uh, try to really enjoy the time that you have in the lab. And now what I can do is I can take questions.
Well, first of all, thank you so much. That was great.
Mhm.
I know we had some questions that people had submitted earlier, so I don't
Yes,
know if you want to maybe start and just sort of read a question and.
let's do that. Um, so I can start with the ones I see right now. So the first question is, in the current job market, what are companies in your field looking for in terms of soft skills? With the advent of AI, some of the computational leverage that interdisciplinary people had may now be considered less valuable. Okay, so I think the question is what kind of soft skills are most companies looking for? Um, I think I can't speak for most other companies, but what I can say um, in terms of what kind of people do you value working with? So, who are the best kinds of people you would want to work with is kind of is how I would interpret the question, and I would say it would be good communicators, A. So, people who really understand the problem, try to they try to bring in the entire team together and understand with them and collaborate with them and are then able to communicate this to a whole other audience. I think that skill is extremely valuable. Uh, and I understand that with some of the computational leverage, I think that that was the second part of the question, that some skills might be less valuable. I'm not sure about that because I think there's a lot of uh, most companies are using AI. They don't use it to review or finish their work. They use it to make it faster. So the processes that were slower are now faster, but you still have to do all the thinking. You still have to do all the verification and the validation. So I think good communicators, A, are highly valuable. Second, I would say someone who's very uh, thorough in their approach, who know how to explain the steps that they took to come to a conclusion. So essentially, the way a lot of communication happens in the industry is that you would say, okay, here's what I did, here's what it led to, and then you explain the why, as opposed to kind of in the past when at least when I was a PhD student, I used to talk about the how first and then why. So I think the framing is important. I think So I hope that answers that question, and I can move on to the next one. How would you apply to the position you are in right now in this job market? That's a that is a good question. So I applied for my position in 2023, and it was kind of on LinkedIn. And I was the I think at the time I also did not see as many positions who that really fit my profile. So it was not that I had too many options. And it really, I think I would, the first step would be through LinkedIn, where you have most of the jobs that are open right now. And you also can reach out to your network who are doing the kind of work that you're interested in. Those are some good ways where you can try to learn about the jobs that are available and what is it that you can improve upon based on your profile. And then the next question is, how much of your PhD do you actually use on the job? Does the title allow you to progress vertically, or is office politics still a limitation? I think so. How much of your PhD do you? I think what I showed today was pretty much most of it, like the thinking, the deep thinking, to be able to zoom in and out, to look at imaging data, what the imaging data means, what could be the sources of artifact in your imaging data, how could you interpret it properly. So I would say a lot of my PhD is what I use in my current job, because it uses pretty much all of the skills that I learned. Um, and does the title allow you to progress vertically? I think this depends on what kind of field you're in and what your interests are. It does in my company. You can progress vertically, and it really depends if you want to progress vertically and manage more people, or if you want to uh, get a wider depth of the concepts. So you could work in, let's say, laser micro dissection as well as spatial biology, so a broader portfolio, or you can rise higher up in spatial biology. And then the next question is, what's the expectation of a, oops, sorry, I missed it. What's the expectation of a PhD hire? At what point do you transition to managing projects, if at all? Um, so usually I would say when you're hired, the first few months are just training. So you do learn everything from scratch. Um, Pretty much everything that you'll use in the job, you get trained on that. And at that point, I think the expectations of a PhD student is to, not a PhD student, a PhD hire is to be able to have that um, ability to think deeply, and ability to manage your time when you're assigned a project, and also to be able to talk to a wider audience. So I think, at least for my role, that was the expectation. And you do get to manage projects. It does come after the immersion time, once you're fully trained on all the techniques that are used in your day-to-day. Then the next question is, some people say that a career path in academia offers more liberty to do research you personally want to do than a career path in industry. Would you agree with that statement? Yes or no, and why? Um, I think it really depends on the environment. I would say with it depends on, yeah, it really depends on how big, let's say, your industry team is versus, let's say, how many resources your academic lab has. because it is a combination of the type of resources you have access to. So I would say in academia, it did feel like you could have more independence to start the project immediately. In industry, you might need to get the buy-in from the stakeholders before you begin a big project that requires a lot of resources. So it really have to talk in terms of resources, as in uh, what all would I need to run this experiment on 20 tissues with 30 markers, those kinds of questions. So I think it could be, it really depends on the size of the lab and your team. And then next question is, generative AI tools are becoming more and more popular. As someone who works with AI methods for analysis, do you have any advice for researchers who are looking into implementing generative AI tools into their workflow? Um, I think I would say AI can be useful if it's accelerating the workflows that you've created. Again, I would say you should always validate, verify, check that the results that it's producing are correct. And two, most importantly, you should have a very clear idea of why you're using AI. So if you're just randomly putting in data and asking it to make predictions with no hypothesis, I would say that that is not a good usage of the tools. Because then you're going from fast to nowhere. And without checking and verifying, you're also not getting anything any faster. So I think you have to be very careful with how you use these tools. That's what I would say. Because if you have a very clear hypothesis, then I think it can be useful to accelerate your research. And uh, given also, this is assuming you're verifying everything and that it's correct, because that can also be a challenge. And then the next question is, when you were considering your next career steps as you finished your PhD, how did you decide between going into industry or continuing in academia? And do you think one day return to academic research? I think that's a really good question. So I would say I was extremely confused when I was doing my PhD, and the slides that I showed, they were kind of trying to show you that, okay, here's what the tools that I used, and this is what these are the programs at Northwestern that really helped me understand at that time in my life what was important to me. So I think at the time, uh, going into the industry made sense. Uh, Again, I think my visa was also a big topic that I had to consider while making that decision. If you don't have that restriction and you're more open, uh, then you have more options as well. So I think you have to factor in a lot of uh, uh, logistics as well. And then to answer the second question, and do you think one day you'll return to academic research? We'll have to see. We'll find out in a few years. Um, but never say never. And then the next question is, what are the pros and cons you've experienced in industry versus academia so far? In general, um, I think for me, one of the pros of um, industry was that it was very efficient, like things get done, and there's always a timeline for every project. There's a risk mitigation strategy for every project. So it's very well planned. So I really like that part of the industry. I would say um, a con would be for me, especially coming from academic background, was I wasn't used to doing work like this at this speed. Because I think the academic work, at least the work that I was doing, was more deeper, so it required a lot more explorative research than very directed work that I could do very quickly. So I would say those were the pros and cons for industry, and then for academia, the pro would be it was, I think, the I really miss working very late in the lab and discussing my results. It's really funny, but I think there was so much learning and so many aha moments that happened, that I think the academic environment really got out of me. So I miss that part. The cons again would be I think that was partly on me. I used to work really long hours because my experiments were long. So I think it was just a consequence of the type of tough experiments I was doing. So it really used to make me tired. Yeah, I think that would be like a very honest uh, answer to this question. And then next question is, what differences have you noticed in how research and teams work since moving to industry? Um, I think it's very similar, at least when you're discussing your results. You do go through weekly meetings to discuss your results, um, similar to academia. Um, and then you make a plan based on the results. I would say there's a very heavy emphasis on making a roadmap before you start a plan. So you make the plan for the whole year, which I don't think was true for academic research because it was so um, sometimes it really biology is like harder to control. So it's hard to know if you'll be doing this experiment like 6 months from now. So I think that is a huge difference in the way of working between academia versus industry. And then next question is, okay, so I see a question from Carla. It says, I want to ask you, how did you approach and prepare for different jobs you applied to and interviews you received? So I think the way I did it was I started by talking to my mentors or uh, members from our lab who had graduated, who were in different roles. I used to have very detailed conversations with them about what their day-to-day is like, what do they like about their job, what excites them the most. That kind of gives you an idea of what kind of work is done in that specific industry. And if you're still in that phase, let's say, where you're deciding which field you want to get into, that's also okay. You can talk to everyone in those different fields, and also go through the externship program, like I said, because that's a good way to meet an even broader audience. And then also asking those questions that are still confusing to you. Certain companies might have a process that requires a referral to get an interview. So I think that's also important to know if that's the path you should be going through. And I think LinkedIn was where I got my current job. I'm not sure about other avenues, but uh, LinkedIn is also a good way to schedule informational interviews with different people who uh, whose work inspires you. So I would start from there and then um, really try to understand what kind of people are they looking for? What kind of skills are they looking for? What is it that you can build upon as you're doing your doctoral work? So I think that kind of would give you a sense of uh, if there's an alignment between your skills and what the job is looking for. I think those are all the questions in the chat. I hope I answered them properly. And if you do have more questions, feel free to I would say maybe LinkedIn might be the best way to connect, just because I don't check my Northwestern email, and I think that might be gone in some time after you graduate. So I would say send me a message on LinkedIn. I'm happy to talk more if you're having those kinds of questions, and even just about science. I'm always happy to chat more about that.